Welcome to the next lecture on protein-DNA interactions. So the major learning objectives of this lecture are basically to learn the major steps of the ChIP-seq protocol, learn what the limitations of ChIP-seq are, learn why we need control experiments when we do ChIP-seq, and most importantly, when it comes to the analysis of ChIP-seq data, one of the major applications of ChIP-seq is to learn where transcription factors bind along the genome in vivo. And so ChIP-seq data is oftentimes also used to gain insight into what the sequence binding preferences of each TF are. And so to really answer the question about what DNA sequences does a TF bind, you really have to be able to find some way to measure or look at, say, for example, an alignment of binding sites of a particular transcription factor, and ask the question, what aspects of these binding sites is different from what I expect by chance, or different from the rest of the genome. And so there's going to be a certain amount of material related to measuring, like, how much information is there in an alignment. How do we measure information, and how do we use that to describe the binding preferences of a TF? So just as a super brief overview, recall from the review lecture that the core promoter that is near the first exon of protein coding genes anyways is generally speaking sufficient for initiation of sort of baseline transcription. It's obviously orientation dependent. It depends on which strand. Depending on which strand the promoter is on, it will yield transcription of different target genes. In terms of transcription factor binding sites, they are located in the proximal promoter, in the core promoter, but oftentimes they're also located far upstream or far downstream of the target transcription start site. And so they can be both enhancing and repressing of target gene expression. They're oftentimes, as we mentioned before, orientation independent in the sense that they can sit on either strand and still function and target the same gene. So methods for measuring TF-DNA interactions fall into two broad categories, the in vivo and in vitro approaches. And so the goal of the in vitro approaches is generally to more quantitatively characterize TF-DNA binding events compared to in vivo. And so with the in vitro methods, you can try to estimate like TF consensus binding sites. You can actually characterize their binding energy landscapes or the actual biophysical parameters that govern the binding events. The in vivo methods are they're more qualitative in the sense that again, you can kind of make a good educated guess about the consensus binding sites of TFs. You can identify obviously where the TFs bind in vivo, and so you can get some idea of sequence specificity and the context of sequence-specific interaction. So you can start to guess as to what other TFs might either cooperatively or antagonistically interact with your particular TF of interest. But again, the in vivo methods are more qualitative in the sense that you can't really estimate these biophysical parameters of these binding events. And so in terms of this course, we're mainly, for the in vivo methods, we'll mainly talk about the ChIP-seq method. It's briefly worth mentioning that techniques like DNase-1 hypersensitivity, depending on how you use it, don't necessarily tell you exactly where one specific TF binds, but they tend to point to regions that are, for example, just generally open, so open chromatin regions. And so generally speaking, transcription factors can only recognize their cognate sequences if they're generally in like open chromatin. And so using techniques like DNase-1 hypersensitivity, you can identify regions that are more likely to be bound by TFs than others. In terms of the in vitro methods, today we'll primarily talk about select-seq and protein binding microarrays, but it's worth noting that there are kind of methods that are still give some kind of like reasonable throughput, like matomy, that we won't talk about, but we'll also briefly mention the electrophoretic mobility shift assays, because those are kind of the most historic methods that were used to characterize TF-DNA interactions. And so, on the other hand, you have kind of, for lack of a better word, you have modern in vivo techniques like ChIP-seq, whose primary goal is to, again, identify in vivo DNA sequences bound by particular transcription factors in this case. And so the general idea is that in vivo in your cell, at any given time, you might have different transcription factors and other factors binding to their target DNA sequences on the genome. And so the idea is that you can add chemicals like formaldehyde, which will basically cross-link proteins or other transcription factors to their target DNA sequence. The idea being that, of course, since transcription factor DNA interactions are transient, by cross-linking, you're basically fixing them in place so that TFs aren't really moving around, and so you're really able to capture that TF-DNA binding interaction. And so what happens after cross-linking is that then you fragment your genome. And so in theory, what you're going to end up with is a bunch of fragments of free DNA, and you're going to end up with a bunch of fragments with your transcription factors bound to their target DNA sequence. And then what's going to happen is that using an antibody, typically like a monoclonal antibody that you've made to target your particular transcription factor of interest, you can pull down DNA fragments that are bound by your transcription factor of interest, which you can then basically sequence, purify and sequence. And then what you do then is what you should end up with is a bunch of reads, which correspond to regions of the genome that are proximal to where your transcription factor bound the genome. And you can take those reads and align them against the genome, and then basically do what's called peak calling, which is basically the task of identifying where across the genome you saw a lot more reads that align to that region than you'd expect by chance, which then suggests that your particular transcription factor of interest is binding to that particular peak or that region in the sample that you IP'd. And so afterwards, so then you can basically do one of two things. Number one of which is through the peak calling process, you've basically identified regions across the genome where your TF is bound, which is great. The second thing you can do is if you want to try to qualitatively and semi-quantitatively characterize what are the general DNA binding preferences of your TF, you can take all of the different peaks conceptually across the genome that you found your TF to bind to, and you can basically align those peaks to each other. And you can basically build a big sequence alignment shown here on the right, where basically all of what you're trying to do with this multiple sequence alignment is try to identify what the transcription factor's particular preferences for different bases are in different positions of its recognition site, essentially. And so ChIP-seq is a super popular way of measuring TF-DNA interactions in vivo. It has a number of limitations, namely that this whole procedure basically relies on your ability to make or buy a highly specific antibody that is both specific and efficient at capturing your transcription factor of interest. Traditionally, you typically needed a lot of IP'd DNA in order for ChIP-seq to work. And so you typically need to perform ChIP-seq on like millions of cells in order to get enough genomic sequence that's bound by your TF of interest in order for ChIP-seq to work. Another major limitation is that oftentimes transcription factor binding is very context dependent. And so certain transcription factors, for example, will only activate under certain environmental conditions or stimuli. And so depending on what state your tissue or cells are in when you perform the cross-linking, you may or may not have subjected those cells to the right conditions in order to give rise to the binding events that you might be looking for. And so you have to be very careful about, there's no such thing as just, capturing all binding sites of a given transcription factor in a given type of cell at once, oftentimes to really get a comprehensive view of where a TF binds in the genome even for one particular type of cell. You'd have to technically do ChIP-seq experiments on many different conditions or under many different stimuli of the same cell type in order to really capture where that TF binds and interacts with in different conditions. And so ChIP-seq is a super popular way of measuring TF DNA interactions in vivo. It has a number of limitations, namely that this whole procedure basically relies on your ability to make or buy a highly specific antibody that is both specific and efficient at capturing your transcription factor of interest. Traditionally, you typically needed a lot of IP DNA in order for ChIP-seq to work. And so you typically need to perform ChIP-seq on millions of cells in order to get enough genomic sequence that's bound by your TF of interest in order for ChIP-seq to work. Another major limitation is that oftentimes transcription factor binding is very context dependent. And so certain transcription factors, for example, will only activate under certain environmental conditions or stimuli. And so depending on what state your tissue or cells are in when you perform the cross-linking, you may or may not have subjected those cells to the right conditions in order to give rise to the binding events that you might be looking for. And so you have to be very careful about, there's no such thing as just, capturing all binding sites of a given transcription factor in a given type of cell at once, oftentimes to really get a comprehensive view of where a TF binds in the genome even for one particular type of cell. You'd have to technically do ChIP-seq experiments on many different conditions or under many different stimuli of the same cell type in order to really capture where that TF binds and interacts with in different conditions. And so some of the problems, again, that you have with using antibodies in the ChIP-seq protocol is that, number one, it turns out to be really hard to get a highly specific antibody because, for example, a lot of transcription factors are part of larger families of transcription factors, or they may share, for example, DNA binding domains that are highly similar with other unrelated transcription factors. And so part of the problem is that you can develop an antibody against your transcription factor, but it can be hard to develop one that doesn't also cross-react or recognize TFs that are not of interest to you. If you actually can't, produce a very good, for example, monoclonal antibody against your TF, there are alternatives. And so, for example, you can basically fuse a tag, such as the HA or FLAG or V5 tag, to your transcription factor of interest. And the benefit of that is that there are antibodies that can recognize these different tags or epitopes that you fuse to your TF. Part of the problem with going down that road is that you can have kind of unintended kind of crosstalk between the epitope that you fuse to your transcription factor of interest, which therefore leads to, for example, altered function or 3D conformation of your protein. And also, in some cases for in vivo studies, obviously it becomes harder to make fusion proteins, for example. Another consideration is that, again, you have to kind of decide, do you want to use monoclonal versus polyclonal antibodies? And I think from a practical perspective, part of the problem with using polyclonal antibodies is that basically there's a lot of kind of technical variation between batches of polyclonal antibodies. And so generally speaking, monoclonal antibodies have less variation from batch to batch, and therefore have less data artifacts that you have to consider downstream. So besides the challenge of acquiring a good antibody against your TF of interest, Some of the other challenges that you typically have to overcome are the amount of genomic material that you need to do a ChIP-seq experiment. And so typically, depending on the concentration of your transcription factor and how frequently it binds across the genome, you might need on the order of like 1 to 10 million cells is input into your ChIP-seq protocol in order to successfully get enough reads to identify the binding sites. And the other typical problem is you need to decide what kind of control experiments you want to run. And so there's typically two different types of controls you would run with a ChIP-seq experiment. You might either use, for example, a non-specific antibody, or you might just use general chromatin input. And so one of the challenges of using, for example, non-specific antibodies are that you typically tend to pull down less DNA than specific antibodies. And so then you would therefore need to do some amplification of your so-called target genomic sequences, which then leads to, again, potentially amplification bias. Some of the reasons you need to do the control experiments are that, number one, it's in terms of the fragmentation step of the ChIP-seq protocol, not all regions of the genome are equally likely to be fragmented. So for example, open chromatin tends to be easier to fragment than others. Again, due to like sequencing bias or amplification bias, during sequencing, there's variable sequencing efficiency across the genome. And finally, some transcription factors or certain domains present in transcription factors are easy to be picked up by antibodies that are not designed to specifically target them. And so even if you think your monoclonal antibody is highly specific, it may not be. And so it may be pulling down some accessory factors, for example, that you need to pick up through the use of like non-specific antibodies as a control experiment. And so getting back to this idea that both the in vitro and in vivo assays both give you a set of both bound fragments and in some sense unbound fragments, or at least control fragments, then one of the main questions that you can ask yourself is, well, what can you really learn from ChIP-seq experiments? And so given on the previous slide that I mentioned that scaling up, particularly in vivo, but also in vitro ChIP-seq experiments to a lot of factors over a lot of cellular contexts is pretty hard and can't be done in the at least short-term near future. One of the primary goals of performing ChIP-seq experiments, one of them anyways, is that we would everybody basically generally wants to be able to build a model that in some sense can accurately describe the DNA binding preferences of a transcription factor. Because imagine that you could, for example, just perform a ChIP-seq assay for a given factor of interest in a few cellular contexts, and then learn some kind of model of how that TF recognizes its cognate binding sequences across the genome. And then imagine that you could actually just take that model and then predict where that TF is going to bind in many other cellular contexts on a computer instead of having to do the ChIP-seq assay. If you could build that kind of powerful model, then that would kind of help you avoid the need to actually perform ChIP-seq assays in all the different cellular contexts that are possible. And so just to give you an idea, depending on how you define cell type, in the human body alone, there could be thousands of different types of cells, never mind all the different contexts in which those cell types can be activated in different ways. And so you can just that should hopefully give you a sense of, how many types of cellular contexts you would actually have to do ChIP-seq assays in to really understand how gene regulation operates in even humans in a wide range of contexts. And so a related question is, whether you use the in vitro or in vivo methods to identify bound fragments of a particular transcription factor, one problem that I'll be skipping over here in this lecture anyways is basically the question of how exactly in a bound fragment in a genomic region which you pulled down from ChIP-seq, for example, how exactly do you find the precise binding site of that transcription factor? Because the binding site of the transcription factor quite often is only like 6 to 8 base pairs, but the fragments that you pull down from, say, ChIP-seq experiments tend to be like, say, 150 to like 200, 300 base pairs. And so how exactly you find the true binding sites of the TFs within each genomic fragment is also a problem on its own, but we'll kind of skip over that detail for now. But suppose that you are able to, for each bound fragment, suppose you're able to identify precisely where that transcription factor is bound, and you were therefore able to perform an alignment to then line up the corresponding positions of each TF binding site in an alignment, like I've shown you here on the left, in the table labeled bound fragments. And so towards building a model of trying to understand how DNA binding works for a particular transcription factor, one important question to ask is, well, when I look at this alignment of bound fragments, how do I determine what the sequence specificity of that TF is at that particular position? I could imagine that given how TFs bind to DNA, I could imagine that some positions are very important to TF DNA recognition, and therefore there should be strong specificity of the TF towards a particular base or a set of bases, that position. And there might be positions where they're not so important for at least direct TF DNA contacts and recognition, and so they're less important. And so then that begs the question, well, how do you define importance? Right? And so one common way of defining importance is looking at a column in this alignment and asking the question, given all of the bases that I see in this column individually, is the distribution of bases, so the percentage of bases that were A versus C versus C versus C, G versus T, how different or surprising is this? Are the representation of these different bases relative to what I expect by chance? And by expect by chance, I mean, more specifically, if you were to look at the control fragments, which represents kind of your background expectation of what kind of fragments are not identified by your particular TF of interest, what bases do I typically see in these control fragments by chance? And so the idea here, the intuition here anyways, is that the more surprised we are about the bases that I see at a given position in my alignment of the bound fragments, the more information there is in that position, or the more important that position is. And so just to give a bit more of a concrete example, if, for example, in the control fragments that I pull down, if on average every base is found at 25% frequency, and then at a particular column in my alignment, if I see all T's, that's probably a strong statistical evidence that at that particular position in the binding site, a T is preferred, probably because of some kind of TF-DNA recognition constraint. So for the next few slides, we're going to discuss the concept of relative entropy, which is one pretty common way of measuring for a given position of your transcription factor binding site within, say, an alignment that you've done between the bound fragments of a TF. Across that single column, relative entropy basically calculates how different is the distribution of bases, so what proportion of the bases you saw were A's, C's, G's, or T's compared to that you saw from the control fragments. And so we'll also discuss the concept of entropy, which is related in the sense that entropy calculates, in this case for a given column of your alignment, how random is the choice of a base for any given sequence within that column. And that entropy calculation is again kind of calculated on a per column basis from your alignment. And so to get you guys motivated and thinking about what it means for a particular DNA fragment or sequence to be random, and therefore, motivate our discussion about entropy. Here on this slide, I've given you guys an example of 4 different short DNA sequences. What I want you to do is actually just take a few minutes to try to write down some kind of shorthand notation for each of these sequences. Try to write down some kind of code that is a kind of a shortest description of what that DNA sequence represents. And so to give you an example, the first sequence is basically 8 adenines stuck together. And so one example of a code you could write, for example, is just A times A, which is another way of just saying A was repeated 8 times. And so I want you to try to come up with a different code, or in a similar format, I want you guys to come up with a code that you could use to write down the remaining 3 DNA fragments. So hopefully after the last few slides, you've got some sort of idea of what it means for DNA sequence to be random. And so there's a few common ways that you might think about what it means for DNA sequence to be random. So the first of which is you might think it intuitively means a sequence is unpredictable in the sense that the past doesn't predict the future. And so if you think about, for example, like a poly-A track sequence, if you have a poly-A track sequence that has, for example, like 1,000 adenines in a row, then essentially after you've seen the 500th adenine, if I asked you, well, what do you think the rest of the sequence is going to look like? You'd probably say, well, there's probably going to be another 500 adenines, and you'd be right. And so in that sense, a poly-A track is highly predictable, and so it's not random. Whereas, for example, if you just, if say, for example, you were making up, if you had decided to make up a random DNA sequence, and suppose when you make up this random DNA sequence, for every base that you pick, you just randomly pick one of the 4 bases at, with equal probability for each base in a whole sequence, then it's unpredictable in the sense that even if you saw the first half of the sequence that I generate, it wouldn't really allow you to accurately predict what the exact sequence of the next half would look like. Another way of thinking about randomness of DNA sequences is that random DNA sequences are not efficient to, you can't really efficiently come up with some way of writing it down easily, right? And so again, if you think back to the case of a poly-A track, if I gave you a DNA sequence that was 1,000 adenines long, and I asked you to come up with some kind of efficient way of writing it down, you would probably come up with something like the phrase or the term 1,000 times A or something to represent the fact that there's 1,000 adenines. But you wouldn't be able to come up with that easy of or that short of a phrase or a notation for a completely uniformly random sequence. Another more subtle but more kind of accurate way of thinking about random sequences is that random sequences essentially have a lot of have more information in them, in the sense that in the same way that a poly-A track, if you've seen 500 adenines, seeing 600 of them, 600 adenines in a row doesn't tell you a lot more than seeing 500 in a row. In the same way, random sequences are a little bit more informative in the sense that even after you've seen 500 randomly chosen bases, that doesn't tell you a whole lot about the precise sequence of next 500 bases if everything is chosen really equally at random. And so random sequences have more information in them. And finally, so the question about entropy really comes back to the question of, well, how random is the sequence I'm looking at, or how unpredictable it is. And so that's in essence what the entropy calculation that you were making before really measures, is just how random or unpredictable your sequence is. All right, and so here's basically the equation for calculating entropy for a single position in a transcription factor binding site alignment. right? So the calculation of entropy is pretty straightforward. What you do is that for a single column in your transcription factor binding site, you look at what fraction of the sequences in that alignment have an A or a C or G or T, right? And so basically for each base, you're essentially asking, if you were to pick one of those sequences at random for that position of the transcription factor binding site, what's the probability that you would have seen an A, C, G, or T, right? And so those probabilities should sum up to 1. And so to calculate entropy, basically for every base, you just multiply the probability of seeing that base by the logarithm of the probability of seeing that base. You add those up across the 4 bases, and then you just negate it. And that's basically your entropy. And so shown on the bottom of the slide is an example of calculating the entropy of an 8-base DNA sequence. And so what you'll see is that in this 8-base DNA sequence, AGCT, TGAC, every base actually appears exactly twice. And so what that means is that every base occupies about 25% of the input DNA sequence. And so if you look at the entropy calculation, basically because every base is present in 25% of the sequence, basically your P log P calculation becomes 2 over 8, or 25%, times log base 2 of 2 over 8. And if you work that out, that basically becomes -½ and -½ times -4 is just 2. And so what I want you to do is following the same equation for calculating entropy, I want you to compute the entropy of these 2 following sequences. So one of them is AAAA, so it's a poly A sequence. And the other is AT. So it's essentially a poly AT sequence. So I want you to compute the entropy of those 2 sequences, see the number that you get, and then I want you to compare it to the entropy value of 2 that we got on the previous slide for AGCT, TGAC. And I want you to take a look and see which sequence got a higher entropy value. So again, entropy in some sense is a measure of randomness, so higher value should mean more like a sequence is more random. And I want you to convince yourself that those numbers make sense, that then the sequence that has the highest entropy value actually does look more random than other sequences that have lower entropy value. So now that you have a sense of what entropy is and how to calculate it, we'll now jump to the topic of relative entropy, which is a way of basically calculating for a given position in a transcription factor binding site, how similar are the distribution of bases that I see in that single position of for example, the sequence alignment to the control or unbound fragments. And so essentially, like, how informative is that position, or how important is that, or is the base that I see at that position, as measured by its difference to what your average column looks like in the control fragments. And so, again, keep in mind that for the purposes of these slides, when I talk about how often we see either an A, C, G, or T at a given position, we're really talking about just considering the set of bases that I see in a single column of an alignment of TF binding sites. Right, and so the general goal here of relative entropy is that we're really trying to compare a model of DNA sequences that we see from a single column of aligned TF binding sites that you got from, for example, ChIP-seq experiment, to some model of DNA sequences from the control fragments to see how similar they are. And so the underlying assumption here is that for positions in a TF binding site, they're important to that particular TF's binding, or that TF DNA recognition. Those informative or important positions are not likely to look like control fragments, control DNA fragments. And so, again, the general assumption is that if a TF is recognizing a specific base of its binding site, if it's very sequence specific, then it shouldn't look like the control sequences that are unbound on average. And so relative entropy is also sometimes called KL divergence. And again, is just a way of measuring how different is a position of your TF binding site alignment to your expectation as defined by the control data. And so the most basic so I've been talking about models of, sequences from your ChIP-seq experiment from one column of that alignment anyways, and models of sequences from the control fragments. And so what I mean in the context of this lecture anyways by a model of sequence is that basically if I look at one column of an alignment of TF binding sites, I can basically just calculate how often or with what frequency do I see a given base in that column of the alignment. So just like with the entropy calculations, for a given position in my alignment of the TF binding sites, I can ask the question, okay, what fraction of the bases were A versus C versus G versus T? And that basically forms my so-called foreground or my model of DNA sequences from my ChIP-seq experiment at a given position. And so here I'm basically calling that model or that distribution of frequencies basically P1. And so the difference between relative entropy and entropy is that relative entropy also requires you to have a model of basically the control fragments. And so in this case, we're going to use a simple model where across all of the control fragments that were pulled down by the control experiment in ChIP-seq, for example, we're going to just calculate the frequency of how often I saw each individual base, so each A, C, G, or T, across all the control fragments. And those frequencies are going to form the set of probabilities labeled P2 here. And so both for P1, the foreground, and P2, the background or the control sequences, I basically have four numbers which give me the frequencies or the probabilities of seeing each of the four bases. And so the equation for computing relative entropy is shown at the bottom of the slide. And so it should look basically pretty similar. It should look pretty similar to the entropy calculation, although there are some differences because there's P1 and P2 instead of just one single set of frequencies. And so just to give you a walkthrough example of how relative entropy is calculated and just to give you a sense of what the number actually means in practice, let's consider an example where, again, we're looking at a single column of the alignment of TF binding sites from the foreground, so from the bound fragments. And suppose that in that column, we see the bases AT, 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 right? And so in that column of the TF binding site, basically I saw an A half of the time, and I saw T half of the time, right? And so therefore, in my foreground model, my P1 model, the probability of seeing an A was basically 4 out of 8 or half. And the probability of seeing a T was 4 out of 8 or half. And the probability of seeing a G or a C was 0 because I didn't see any in that column. Right? And so let's assume for the time being that when you look across all of the control fragments, basically we saw all bases with equal frequency. So all bases were present in 25% each. Right, and so in terms of the relative entropy calculation, basically, again, it looks like basically for relative entropy, we are looking at each base individually, and we're just asking, okay, what's the probability of seeing that base in my given column in my bound fragments? Then I multiply that by the log, now it's a log ratio of the probability of seeing that fragment, or seeing that base in the control fragments divided by, again, the probability of seeing that base in a particular column of the bound fragments. And so this is basically what the calculation comes out to. Basically it comes out to 1, a value of 1. And so something important to note when you calculate relative entropy is that 0 times anything is still considered 0. So even 0 log 0 is equal to 0 in this case when you calculate relative entropy. And so the question you should be asking yourself at this point is, what does the relative entropy of 1 mean? And so it's kind of beyond the scope of the class to really define what the actual relative entropy number means. But what's more important here is really just understanding what kinds of sequences get higher relative entropies than others. And so let's assume again that from our control fragments, we saw that each base was present in basically equal proportion. So every base was present in about a quarter of the sequence. And so what's kind of interesting is to think about what the relative entropy of a sequence that also has an equal representation of all bases. And so say, for example, we were looking at a column of the transcription factor binding site alignment, and we saw the bases ACGT, ACGT. And so all bases are also present in 25% of the time. If you work out the relative entropy calculations, which I encourage you to do so, then you'd find that the relative entropy is actually 0 for ACGT, ACGT. And so on the previous slide, which was a poly AT sequence, we saw that the relative entropy was 1. And so basically the point here is that the poly AT sequence has a higher relative entropy compared to ACGT, ACGT, because the base frequencies are more different from the control fragments, right? And so the take-home message here really is that when you calculate relative entropy, if you see a large positive value, that means that in your column of the TF binding site that you're using to calculate P1, it's more different from your model of the control fragments. And so it's because it's different from the background genome, then it's surprising. What you're seeing is surprising, or it's informative, because basically it's suggesting that at that particular position of the TF binding site, your transcription factor has a strong preference for bases that you don't typically see in the rest of the genome, or at least in the control fragments. And so that position is more informative. Whereas if you get a relative entropy of 0, that means that at that particular column of the TF binding site, you're seeing exactly what you'd expect to see, at least based on the control fragments, your model of the control fragments. And so what you're seeing there is unsurprising, or it looks just like the rest of the genome. And so the underlying assumption there is that there's no detectable sequence specificity of the TF at that position of the alignment, at least according to your model of TF binding sites. And so that pretty much concludes our discussion about relative entropy and trying to detect which positions of a TF binding site are incur sequence specificity of the transcription factor. What I do want to say is that in real life, people oftentimes try to use more realistic models of TF binding, either TF binding or the background control fragments, because making the assumption that treating each column of the alignment or the transcription factor binding site as being independent is a fairly poor model of transcription factor binding, because we know that that a lot of positions have codependencies between them. So transcription factor doesn't just use each base at each individual position independently when it recognizes it for binding. There are dependencies between the positions when you look at how transcription factor binding sites differ between different sites. And so we won't really talk about what those more kind of realistic models of TF binding are, because they are a little bit more complicated. But I do want to leave you with an idea of what more complicated models might mean in kind of a more intuitive, hopefully intuitive example. And so at the beginning of this section of the lecture, we talked about the randomness, how to measure randomness of DNA sequences. And one of the statements I made was that sequences which are predictable tend to have less entropy, right? Because you can kind of guess, based on reading, for example, the first half of a DNA sequence, you can kind of guess what the second half will look like. And if that's the case, then that sequence is less random because it's predictable. And so most of the time when we are looking at, if you notice carefully, most of the time when we were looking at sequences that were predictable, we were mainly looking at like repetitive sequences, like things like poly-A tracks. Poly-A tracks are, highly predictable and non-random because, if you see like 20 bases in a row that are all A's, then the chances that the next 5 are going to be all A's is, pretty high if you're looking at poly-A sequences, for example. But it's important to note that although a lot of these kind of simple repeat sequences are predictive and therefore less random, not all predictable sequences themselves are repetitive, right? And so if you consider, for example, like a sequence of numbers like the Fibonacci numbers, right? So the Fibonacci sequence basically starts with the numbers 1 and 1. And basically the idea is that the next number in your Fibonacci sequence is the sum of the previous 2 numbers. And so if you look at the sequence 1, 1, 2, 3, 5, 8, 13, you'll see that each number is the sum of the previous 2 numbers in the sequence. And so that sequence there, 1, 1, 2, 3, 5, 8, 13, doesn't look repetitive per se, but it is predictable because once you know the first 2 numbers in the Fibonacci sequence, you can calculate the rest of them. Similarly, if you consider, something a little more related to molecular biology, if you consider protein coding sequences, and you consider, for example, codon usage. So what codon usage means is that even though, generally speaking, for eukaryotes are, the lookup table that you can use to figure out, which triplets basically translate into different amino acids, even though that table itself is relatively fixed across, most eukaryotes and so on, you'll notice that for a given amino acid, there are typically more than one codons that encode that amino acid. And so if you look carefully across different organisms, what you'll find is that certain organisms tend to prefer certain codons more than others. If you consider all the protein coding sequences in a given genome, you'll find that the codon usage pattern for a given amino acid can differ between genomes. And so what that means is that if you are suppose you were trying to build a model of gene sequences, of protein coding gene sequences, if you know if the codon usage of a genome is very skewed, and so suppose that for any given amino acid you only see exactly one codon being used to encode that amino acid in any of the protein coding genes in that genome, then the protein coding model is protein coding genes are essentially more predictable than genomes that use, say, all of the codons for all of the different amino acids equally well. Because, for example, if you see a cysteine, if you know there's going to be a cysteine at a given position in a protein coding sequence, then for the genome with highly skewed codon usage, then you know exactly which codon is going to be there, even without looking at the sequence. Whereas for a genome that uses all of the codons for example, cysteine equally often, then even armed with the knowledge that you're looking at a cysteine coding triplet, then you still don't exactly know what the DNA sequence will look like. And so basically, in short, when people actually consider the relative entropy of transcription factor binding sites of individual columns of the transcription factor binding site, what the relative entropy and how much information is considered to be present at that particular position can actually differ depending on what your model of random DNA sequences based on the control fragments look like. And so just be aware that there's more than one way of calculating what you expect to see at a given position in a transcription factor binding site, depending on how you model the control fragments. So now that we have some basic understanding of what entropy and relative entropy is, it's time to go back to discussing how to represent binding sites for a particular transcription factor that you've identified from, for example, the peak calling at the very end of the ChIP-seq protocol. And so suppose that you have a set of aligned binding sites of your given transcription factor. There's a couple of different common ways that TFBS are represented. So the classic way that was first used is basically what's called the consensus sequence, which is basically just a representation that summarizes the most frequently observed bases at each given position in the binding site. And so you'll notice for the consensus sequence representation, there's a whole bunch of letters that you probably don't recognize, like VDR, TWR, so on and so on. What these are basically new characters that are obviously not real bases, but they correspond to the occurrence of more than one base that is commonly observed at a given position. Basically, IUPAC came up with a consistent set of characters that are used to represent different combinations of bases at a given position. But the consensus sequence approach basically is not really used that frequently nowadays because everybody kind of recognizes that transcription factors can have different levels of sequence specificity at a given position in the alignment. And so, the precise specificity can't really be captured by consensus sequences. And so they were kind of replaced by what we'll discuss next, which is basically the sequence logo and position weight matrix. And so it's basically a precursor to the position weight matrix. What you can do instead of just a simple consensus sequence is that you can look at your alignment of your binding sites. And for every position in the alignment, you can basically calculate the number of times you see a given base at that position. And so that counting basically gives what's called a position frequency matrix, where the rows represent different bases and the columns represent different positions of the alignment. And so you can see that it basically just gives you a quick idea about which base is found, which position in the alignment. And this position frequency matrix is what's used to generate the famous sequence logo. And so the sequence logo, again, is a diagram, a visualization of the position frequency matrix, where columns in the sequence logo correspond to columns in the position frequency matrix. And the total height of a given column represents what's formally called as the information content of that position. And so for the purposes of this class, you can think about information content as basically what we refer to as the absolute entropy calculation. So basically, the height of the column is roughly proportional to how non-random the bases that you observe in that given position of the alignment is. And so the larger that number is, the more strongly the frequencies deviate from equal representation of all bases at that position in the alignment. The height of the individual letters basically, in some sense, corresponds to the contribution of each base to that total information content. So basically, the more a letter, a particular base, deviates from 25% frequency, the higher that column will be, the higher that base will be in the sequence logo in the corresponding position. And so sequence logos are good for visualizing a position frequency matrix and for giving some indication as to what the sequence specificity of the TF is at a given position in the alignment. But actually, more people would use what's called a position weight matrix to actually go and search new DNA sequences to find binding sites of a particular TF. And so the goal of a PWM is to be able to look at a short DNA sequence of length, the same length as the transcription factor binding site. And the goal of the PWM is to basically score that sequence for how likely it is to be bound by your TF of interest. So big positive scores correspond to high likelihood of TF binding that sequence, and small numbers mean that TF is less likely to bind that particular sequence. And so in the middle of this slide, I basically show you how to go from a PFM, or position frequency matrix, to a PWM. And so you can see that a PWM is represented by a table of the same size as a PFM. And basically, the expression in the middle of the slide, log of PBI over PB, is how you go from the PFM to the PWM. And so at this point, I want you to recall our earlier slides about relative entropy. If you remember, relative entropy is basically a calculation about which computes how different the distributions P1 and P2 are from each other. And as you recall at the bottom of the slide, one of the key components of that relative entropy calculation is this log ratio. It was the log of the probability of seeing a character in the control fragments versus log probability of seeing a base in the bound fragments. And it's the negative of that. And so if you look at how PFMs are used to calculate PWMs, basically for a given position and base in the binding site. So for example, if you looked at, let's say, in column 2, if you look at C, basically C is observed, if you look at the position frequency matrix, C is observed 2 out of 5 of the input sequences. And so it's represented, so C represents about 40% of the input sequences at that particular position. And so how you go from, how you calculate the contribution of observing a C at the second position in the sequence is by computing the log of the probability of seeing a C in the second position based on your PFM. So that would be like 0.4 corresponding to 40%. And you divide that 40% by the probability of seeing a C in your control fragments. And then you take the log of that. And so basically that logarithm, that log ratio, is again going to be positive when you see that base more often in your PFM, so in your bound fragments, compared to your control fragments represented by PB. And the bigger the number is, the more positive that number is, the kind of the more deviating your base, the more frequently you see the base in that particular position compared to the control fragments. And so repeating that calculation, you can kind of fill out this PWM table and basically compute a score for each base in each position. And then basically what you do is that once you have a new sequence like TGC, TG, what you can do is you can basically, for every base in your input sequence, TGC, TG, you can look up the corresponding value that that base contributes to that position of the binding site. So basically, if you see the yellow entries highlighted in PWM, that corresponds to the sequence TGC, TG. And when you add up those values that are highlighted in yellow, you basically get the score of that sequence. And so in this case, it's 0.9. And so there's like a slight preference for binding TGC, TG compared to background expectation. And so the idea of detecting binding sites, so suppose you have like a genome, an entire genome that you want to scan for binding sites corresponding to your TF of interest. What you can do is once you have your PWM, you can basically slide a so-called window. So imagine having a window that's the same size as your TF binding site, or the same number of columns as your position weight matrix. And basically as you're sliding this window across the genome, you can basically calculate what the score of your TF is in terms of binding to that particular window of the genome sequence. And then you can basically computes scores, and you can score each window as you move along or scan the genome in terms of, trying to find regions of the genome that have high score. And so typically speaking, when you're scanning a genome sequence for locations of a binding site, you can either look at the absolute score, which is basically what I discussed on the previous slide, where you just kind of add up the contribution of each base to the total score of a window, and that gives you your absolute score. Or what sometimes people do is they compute what's called a relative score. And so for any given PWM, you can easily identify the sequence that will give you the highest possible score, right? And so you can do that by basically looking at each column of your PWM, just checking out which base gives you the highest score, and just making a fake sequence, for example, that has that base at that position. And after you look at all the columns, you'll basically arrive at the sequence that gives you the highest score from the PWM. In a similar way, you can also calculate what the smallest score you could get from a PWM is by looking at the smallest number in every column and just adding those up, and that'll give you a minimum score. And so given the maximum score possible and a minimum score possible, as well as your absolute score that you get from a given sequence, you can use the equation on the bottom right to calculate what's called a relative score, which tells you how, what percent of the maximum possible value does the sequence of, that is contained within a window give you relative to the maximum and minimum that you could have gotten from the position weight matrix. And so in practice, there's a lot of people, a lot of people who spend a lot of time basically building software to, number one, kind of look through the literature, look through ChIP-seq data, through select experiments, and build up libraries of sequence logos, as well as position weight matrices for different transcription factors from different species, and they basically create these big databases of logos and PWMs. And so on the left, I've given you some examples of some of the most common databases that people use for if they want to download like a PWM for a transcription factor of interest and then use it to scan their own genome to figure out where TF binds. And so again, kind of going back to the idea of, well, what can you do with a sequence logo and position weight matrix? So number one, again, you can basically use it to get some sense of the sequence specificity of a transcription factor. And so the sequence specificity of a transcription factor is dictated by how it interacts with both the individual bases that are present in the position weight, or the individual bases that are present at its binding site, as well as the interactions of the TF with the backbone of the DNA as well. And so basically, when you see positions where there's a lot of sequence specificity, that tends to indicate potential, for example, direct interactions between one or more amino acids on the transcription factor to that particular base at that position of the binding site. Whereas sort of less strong preferences for bases could indicate an indirect effect of bases on like the shape or the charge of the backbone, or just might indicate less direct contacts between amino acids and DNA. And so one obvious question is, how good are PWMs for actually finding real binding sites of transcription factors? And so the answer is, it depends on what the context of this binding is. And so if you're just talking about in vitro binding of a transcription factor to, say, naked DNA, the PWM scores actually pretty well correlate with in vitro binding. And so lots of people have done studies trying to measure correlation between the predicted likelihood of binding is via the PWM scores to the actual binding observed in vitro, and found that by and large there's pretty decent correlation between the predicted and actual binding of TFs to DNA. So one of the less popular ways of generating regulatory networks is through position weight matrix scanning. And so we already kind of went through how to construct position weight matrices and how to do a basic scan using them. Here I'll just talk a little bit about pitfalls of using them to generate regulatory interaction networks. And so the first major problem you have is that there's a lot of degeneracy in transcription factor binding site recognition sequences. And so, like I mentioned before with PWM scanning, PWM scanning is good at detecting in vitro binding sites, but not so much in vivo binding sites because there's a lot of in vivo, there's a lot of other factors that influence whether TF binds or not, like whether or not the chromatin is open at that particular position, or whether or not there's other cofactors that are present that facilitate binding. One of the other big problems is that PWMs are not actually unique to any given transcription factor. So lots of transcription factors form families of transcription factors because they have related protein sequence and structure and function. And so here, for example, I'm showing you 3 different transcription factor families and different members of that family, as well as their kind of known binding sites or known position sequence logos. And you can kind of see that different members of the same family can have very similar sequence logos and position weight matrices. And what that means in practice is that it's actually really hard to distinguish the binding sites of these different factors. And in fact, in reality, within a given family, you might have a lot of cross-binding of one transcription factor binding to the binding site of another member of the same family, but just at like different binding affinity. And so, it's really hard to predict differences in binding of factors of the same family. And even factors of different families that share, say, parts of DNA binding domains and so on, a lot of their binding sites look pretty similar. Number 2, there's a big question that we didn't really talk about, which is that it can be challenging to figure out which position weight matrix to use. And so in the ChIP-seq lecture, I basically mentioned briefly that there are many databases out there where people have taken ChIP-seq data or selects data or PWM data and assayed different transcription factors to generate sequence logos. One issue is that there's a lot of kind of technical variability in between the different assays that you can use to generate position weight matrices. Even for ChIP-seq, like depending on which cellular context you ChIP-seq a particular TF in, you might get a different set of binding sites, and that might lead to different position weight matrices. And so here, for example, under number 2, I'm showing you 4 different PWM or 4 different sequence logos and respective PWMs for the same transcription factor, UBX, that's found from different websites. And you can basically see that even though they all kind of qualitatively look similar, if you were to actually use these 4 different PWMs or sequence logos to actually scan and search for UBX binding sites, they would actually return different hits, just because the specificity at each position is actually quite different between these 4 different PWMs for the same factor. Again, number 3 basically refers to the fact that binding in vivo is often context dependent, and PWMs currently do a poor job of capturing that context dependency. Number 4, PWM libraries oftentimes are really incomplete, and so in the human genome, for example, depending on how you count it, people think there's roughly like 3,000 DNA binding transcription factors, most of which we don't have a PWM for in the first place because someone just wasn't able to, mostly because someone either wasn't able to generate a good antibody for each one of those TFs, or or those TFs just haven't been studied yet. So maybe unsurprisingly in hindsight, it turns out that even though PWMs seem to be able to score in vitro binding sites fairly well for transcription factors, it turns out that they don't really predict in vivo binding sites very well. And so the agreement between like a PWM scan of a genome and, say, a ChIP-seq assay, if you've done in a kind of in a naive way, are pretty uncorrelated. And so it turns out that the in vitro predictions made by PWMs tend to overpredict binding sites of transcription factors, whereas in vivo, most transcription factors don't bind nearly as many sites as that predicted by the PWM. And so this isn't entirely surprising because, as I mentioned earlier, PWM or transcription factors require their binding site to, for example, be accessible. And so obviously in a lot of cellular contexts, a transcription factor binding site might be wrapped up in histones or shut down epigenetically. And so even though there is technically the potential to be bound, the physical accessibility of the genome at that location is just such that the TF won't actually bind there in vivo under many cellular contexts.